Welcome back to the deep dive. Last time, we sort of sketched out the nervous system's big map, its main job, communication, and that split, between the central nervous system, the brain and spinal cord,
Mhm.
and the peripheral nervous system, basically everything else reaching out.
Right, the broader network.
Today, we're zooming way in, like microscopic level. We're meeting the neurons.
Fundamental unit, yeah.
Yeah.
The cell that actually does the communicating.
These incredible cells, just built for speed receiving, processing, sending info.
And this deep dive, it really draws from Mr. Hostler's lecture guidance, which is adapted from Chapter 10 of The Human Body in Health and Illness by Barbara Herlihy. So we're focusing on his approach to understanding how a single neuron is built and, crucially, how it fires.
Right. Our goal today is really to break down that structure and function. Think of it as getting you wired into how your own nerves actually work. Okay, so let's unpack this. If the whole system's a network, what's the wire? What does a typical neuron actually look like?
Well, fundamentally, a neuron has three main parts, three anatomical regions. Each has a very specific job.
Like stages in the process.
Exactly. You've got the dendrites first. These are usually short branching bits sticking out from the cell body, kind of like little tree branches.
Okay.
Their job is basically reception. They're the listeners. They pick up chemical messages, signals from other neurons, or maybe sensory inputs.
So they're like antennas, just grabbing signals.
Pretty much. They gather that input and, importantly, carry it toward the cell body.
Gotcha. Which is part two.
Which is part two. The cell body, or soma, that's the neuron's headquarters. It's got the nucleus, the organelles, all the standard cell stuff. But functionally, its big role is integration. It takes all those signals coming in from the dendrites and makes the call. Is this signal strong enough to pass on?
Ah, the decision maker. So this is the go or no-go point.
That's it. And if it's go,
Yeah.
then the signal travels down the third part, the axon. This is usually one long projection stretching away from the cell body, sometimes really long. Its job is transmission, sending the signal away from the cell body. And at the very end, it branches out again, these terminal branches, to connect to the next neuron or maybe a muscle cell.
So the flow is always one way. Dendrites get it. Cell body thinks about it. Axon sends it out.
That's the fundamental direction. Dendrites, a cell body axon. That one-way street is key for keeping things organized.
Okay, here's something interesting, though. Neurons aren't like, say, skin cells, right? They don't just divide and make new ones easily.
That's a really crucial point. Mature neurons generally lose that ability to divide. They're highly specialized for communication, for speed, but that specialization comes at a cost. Nerve damage can be, well, often permanent or very slow to heal because they can't just regenerate easily. It's a trade-off.
Okay, structure and direction makes sense. But how does that decision in the cell body turn into an actual electrical signal zipping down the axon? How does it fire?
Ah, right. This is where the electrochemistry gets really cool. See, even when a neuron is just sitting there at rest, it's not truly quiet. It's polarized. It has an electrical charge.
Like a tiny battery?
Exactly like a tiny battery. It's holding a charge across its membrane. We call this the resting membrane potential.
And what creates that charge?
It's mostly about ions, specifically sodium, Na plus less, and potassium, K plus A. At rest, there's a different concentration of these ions inside versus outside the cell. Generally, it's more positive outside and more negative inside the neuron.
Oh.
And this difference, this resting state, is actively maintained. There's a mechanism called the sodium-potassium pump, constantly working in the membrane, pushing ions around to keep that charge difference stable.
So it's actively holding itself ready, waiting.
Precisely, waiting for enough stimulation. When those dendrites get signals, those signals cause little changes in the membrane charge at the cell body. If enough of those little changes add up, if they reach a certain critical level, a specific voltage, called the threshold potential, then bang, it triggers.
Like flipping a switch. That's the go signal.
That's the go signal. Reaching threshold triggers the action potential. That's the electrical impulse, the nerve signal itself, and it unfolds in a very specific sequence.
Okay, what happens?
First phase, depolarization. Special channels, gates for sodium ions, suddenly open up. Sodium is concentrated outside, remember?
Right.
So it floods into the cell, down its concentration and electrical gradients. All that positive charge rushing in makes the inside of the cell very quickly become positive.
Goes from negative to positive fast.
Very fast. Then, almost immediately, comes repolarization. Those sodium channels slam shut and different channels, once for potassium, open up. Potassium is concentrated inside the cell. So now potassium ions rush out. taking their positive charge with them. This outflow of positive charge brings the inside of the membrane back down towards its negative resting state.
Okay, so depolarization, sodium in, repolarization is potassium out.
You got it. And then there's a brief reset or refractory period where things get back to the initial state, largely thanks to that soda and potassium pump getting the ions back to their starting positions. Ready to fire again.
And that wave, that depolarization, repolarization sequence, that's what travels down the axon.
That is the action potential. It propagates, it travels down the axon like a spark traveling down a fuse.
Wow. Okay, but some signals are incredibly fast. Like you touch something hot and your hand pulls back instantly before you even think, ouch. How do neurons get that fast, especially over, say, the length of your arm or leg?
Yeah. For speed, the key is often myelin, the myelin sheath.
Myelin, I've heard of that. It's like insulation.
Exactly. It's a fatty, insulating layer that wraps around the axon. Think of the plastic coating on an electrical wire. It prevents the electrical current from leaking out.
And where does that come from? Is it part of the neuron itself?
No, it's actually made by other cells, supporting cells called neuroglia. We'll talk more about them next time, but the ones that make myelin are different depending on where you are. In the central nervous system, brain, and spinal cord, it's oligodendrocytes.
Okay.
Out in the peripheral nervous system, the nerves in your limbs, your body, it's Schwann cells.
Schwann cells in the periphery. Got it.
Yeah.
So is the axon just completely coated, like a sausage casing?
Almost, but not quite. And this is crucial for the speed boost. The myelin sheath isn't continuous. There are these tiny little gaps, exposed sections of the axon membrane at regular intervals.
Gaps.
Yeah, they're called the nodes of Ranvier, named after the guy who discovered them, Louis-Antoine Ranvier.
So the insulation has breaks in it. Why?
Because the action potential, that wave of ion exchange, can only happen where the axon membrane is exposed at those nodes of Ranvier. It can't happen under the myelin insulation.
So the signal has to jump.
Exactly. It jumps from one node to the next. The electrical current flows very quickly under the myelin segment to the next node, where it triggers the depolarization-repolarization sequence again.
Wow.
This jumping pattern has a name, saltatory conduction. Saltatory comes from the Latin saltare, meaning to leap or to jump.
Saltatory conduction?
Yeah.
The signal is literally leaping down the axon.
It is, and because it only has to regenerate the action potential at these nodes, instead of continuously along the entire length, it travels much, much faster.
How many faster?
Oh, it can be up to like 100 times faster than on an unmyelinated axon of the same size.
That explains the speed for reflexes then, and feeling things quickly.
Absolutely. That speed is critical for rapid responses, quick reflexes, transmitting sharp pain signals efficiently, and allowing for really precise, fast muscle control. Myelination is key for high-speed nerve communication.
Okay, so let's try and wrap this up a bit. Looking back at Mr. Hostler's guidance on the neuron, what's the main takeaway here?
I think the main takeaway is just how elegantly designed this single cell is for communication. You have the clear structure, dendrites listening, cell body deciding, axon transmitting, that's the path.
Right.
Then you have the electrical process, that resting charge hitting threshold, the action potential driven by sodium rushing in and potassium rushing out, all reset by the pump. It's an electrochemical event.
And then the speed boost.
And then the speed boost, yeah.
Yeah.
Myelin insulation formed by Schwann cells peripherally, or oligodendrocytes centrally, allowing that saltatory conduction, the jumping between nodes of Ranvier. It highlights the system's efficiency. It really is the basic language your nervous system uses. Everything else, all the complex stuff the nervous system does, it builds on this fundamental ability of the neuron to receive, process, and send signals quickly.
Okay, fantastic. Now let's leave everyone with something to chew on connecting these pieces.
All right. Imagine you encounter a patient, maybe in a clinical setting, and they're showing slowed reflexes, specifically in their arms and legs. And let's say tests reveal the issue is damage to the insulating layer around the neurons in their peripheral nerves out in those limbs.
Okay, damage to the insulation in peripheral nerves.
So based on what we just discussed, the structure, the function, the speed mechanisms, think about this. What specific structure is likely damaged on those peripheral neurons? And importantly, which type of supporting cell we talked about is responsible for making that insulating layer in the peripheral nervous system?
Think about the parts, the insulation's job, and which cells operate where. Good one.
We'll actually pick up on those supporting cells next time. On the next deep dive, we're focusing entirely on the neuroglia, the astrocytes, microglia, and we'll definitely revisit Schwann cells and oligodendrocytes. You'll see why neurons really can't do it alone.
Sounds great. Well, thanks for taking this deep dive into the neuron with us today. Hope it gives you a clearer picture of this amazing little cell.